warm welcome to one and all. Today's session is on biosynthesis of fatty acids and its regulation. Myself, Dr. Mausmi Shankaradala, working as an assistant professor in the Department of Biochemistry, St. Joseph's College for Women Autonomous, hereby brings a brief description about what is the biosynthesis of fatty acid procedure and its regulation. The current topic involves following contents: fatty acids introduction, straight chain fatty acids, saturated straight chain fatty acids, steps involved in fatty acid biosynthesis, regulation of fatty acid
Learning
biosynthesis.
outcomes involve biosynthesis of fatty acids, different pathways, and its regulation.
Essential for the structural molecules and energy storage in our body. They are necessary for a number of physiological processes, including the synthesis of cell membranes, cell signaling, production of energy, etc. Many metabolic and enzymatic mechanisms are involved in the complex and tightly regulated process that produces these fatty acids. A complex sequence of biochemical events known as fatty acid synthesis entails the conversion of acetyl-CoA to malonyl-CoA and the successive addition of 2 carbon to lengthen the fatty acid chain in the fatty acid synthase complex, which is abbreviated as FAS complex. Condensation, reduction, dehydration, and further reduction process are involved in this process, which yields saturated straight chain fatty acids. More desaturation process, either aerobic or anaerobic, are needed to be produced to form unsaturated straight chain fatty acids. While tuberculose stearic acid production and omega alicyclic fatty acids production are involved in certain organisms, odd chain and branched chain fatty acids have been distinguished in their routes. The various routes that contribute to the rich variety of fatty acids present in biological systems are crucial for comprehending lipid metabolism, its consequences for health, and possible treatments. Coming to the straight chain fatty acid, as the name implies, fatty acids having a linear carbon chain are known as straight chain fatty acids. They can be further divided into straight chain fatty acids, unsaturated and saturated, according to whether or not their carbon chains include double bond connecting the carbon atoms. Saturated straight chain fatty acids. In saturated straight chain fatty acids, the carbon atoms in the carbon chain do not form double bonds. The process of lipogenesis, which converts the carbohydrates into fatty acids, involves various routes for the production of these fatty acids. The process of lipogenesis occurs in the liver and in the adipose tissue, and therefore there are a number of vital enzymes present in this place. The initial stage of metabolic process that converts carbohydrates into fatty acids is the production of acetyl-CoA. Then in the cytoplasm of the cells, acetyl-CoA is used as a substrate for the production of fatty acids. Fatty acids are produced by a series of reactions that are catalyzed by several elongates, desaturates, fatty acid synthase, and acetyl-CoA carboxylase. Animals cannot synthesize or resynthesize carbohydrates from fatty acids, while they can only produce carbohydrates from fatty acids through a process called as gluconeogenesis. The fact that fatty acids cannot be changed back into carbohydrates emphasizes how crucial it is to keep intake and output of energy under check. Unsaturated straight chain fatty acids. The unsaturated straight chain fatty acids, on the other hand, have one or more double bonds within their carbon chain. There are two different ways to introduce double bonds: aerobic desaturation and anaerobic desaturation. Anaerobic desaturation is the process by which some microorganisms add double bonds to the fatty acids without the presence of oxygen. This process is catalyzed by specialized enzymes called aerobic desaturates. On the other hand, human and higher animals undergo aerobic desaturation, which requires oxygen as a cofactor for the desaturate enzymes involved. The next topic is the odd chain fatty acid. Odd chain fatty acids are a different class of fatty acids with an odd number of carbohydrates in the carbon chain. These fatty acids are essential to numerous biological processes, even though they are not as common as their even chain counterparts. It is possible to convert some amino acids such as valine, isoleucine into odd chain fatty acids. The breakdown of these amino acids results in the production of the fatty acid subsequently converted into odd chain fatty acids by numerous mechanisms involved. Branched chain fatty acids. Branched chain fatty acids, as the name suggests, contain one or more branches in their carbon chain. One special enzyme which is involved in the synthesis of branched chain fatty acid is the branched chain fatty acid synthesizing system. This technique uses an enzyme called branched chain fatty acid synthase to add branches to the fatty acid molecule. In certain bacteria, branched chain fatty acid synthesis is an essential process that influences a variety of biological processes. Coming to omega acyclic fatty acid, a unique class of fatty acids known as omega acyclic fatty acids have a methyl group at the carbon chains omega and at a cyclic structure. These fatty acids, which are found in several marine species, have been found to have health benefits such as anti-inflammatory and cardiovascular protective measures. Enzymatic pathways that create omega acyclic fatty acids are specific and not fully understood. Researchers are continuously searching for the potential therapeutic applications in order to gain further insight into the physiological functions and biosynthesis of these specific fatty acids. Importance of the fatty acid. It has many underlying lipid metabolism requires an understanding of the intricacy involved in the synthesis of fatty acid. However, it has a lot of significant effects on the production of food, medicine, and other business. The etiology of multiple metabolic disorders such as diabetes, obesity, and cardiovascular diseases have been proposed to involve the dysfunctioning of the synthesis of fatty acids. The steps involved are, first one is the priming, which begins with the formation of primer molecule, that is acetyl-CoA. The second one is the malonyl-CoA formation, acetyl-CoA carboxylated to malonyl-CoA by acetyl-CoA carboxylase, requiring ATP and bicarbonate. The third one is initiation, which involves the acetyl-CoA from acetyl-CoA transfer to acyl-CoA carrier protein on the fatty acid synthase, that is FAS enzyme complex, and malonyl-CoA also transfer to another ACP on the fatty acid synthase complex. So coming to the flowchart, here you can see acetyl-CoA is converted into malonyl-CoA with the help of one more acetyl-CoA. Then it results in the formation of 3-keto-acyl carrier protein, which is further converted in the presence of reductase, the 3-hydroxy-acyl carrier protein. In the presence of dehydrogenase, it is resulting in the formation of 2,3-trans-enoyl acyl carrier protein, which on further addition of all the molecules, that is branching, occurs, and it results in the formation of lysophosphatidate, which results in the formation of phosphatidate, diacylglycerol, and triacylglycerol. Here, one of the intermediate is glycerol 3-phosphate, which is formed from the conversion of glycerol. The fourth process in the elongation is the elongation. The main cycle of the fatty acid synthesis involves 4 more reactions in the elongation, that is condensation, reduction, dehydration, and another reduction. Under condensation, acetyl group form acetyl-CoA condenses with the methylene group of malonyl-CoA, releasing carbon dioxide. Under the reduction, beta-ketoacyl group is reduced to beta-hydroxy-acyl group. And then under the dehydration, water molecules removed from beta-hydroxy group, forming a double bond between the second and third carbons. And last one is another reduction phase where double bonds are reduced to form a single bond, resulting in a fatty acyl-CoA molecule with 2 more carbons that are starting molecule. Repeated with another molecule of malonyl-CoA, further lengthening the fatty acid chain by 2 carbons with each cycle. Finally, termination. Elongation cycle continues until a fatty acid chain of specific length of carbons is formed. Finally, fatty acyl-CoA molecule is released with the action of the enzyme thioesterase. Modification. Newly synthesized fatty acids can undergo further modifications such as desaturation and also hydroxylation. The fatty acid synthase complex is a large multi-enzyme complex housing all the necessary enzymes for the fatty acid synthesis. Each enzyme catalyzes a specific step in the pathway, ensuring all the efficient production of fatty acids. The enzyme of fatty acid synthesis, which is also known as lipogenesis, is the process of synthesizing fatty acids from acetyl-CoA and malonyl-CoA in the cytoplasm. This process is catalyzed by a series of enzymes. The enzymes are acetyl-CoA carboxylase, which is abbreviated as ACC. This enzyme catalyzes the carboxylation of acetyl-CoA form of malonyl-CoA, which in the form of first committed step in the fatty acid synthesis. This is regulated by both allosteric and covalent modification. The second one is the fatty acid synthase. It is a multifunctional enzyme complex synthesis of the long chain fatty acids. It consists of multiple enzyme activities, including acyl carrier protein, which carries the growing fatty acid chain, and various enzymatic domains that catalyzes the individual steps of fatty acid synthesis, such as condensation, reduction, dehydration, and further reduction. Thioesterase, this enzyme catalyzes the hydrolysis of fatty acyl ACP to release the free fatty acid. These enzymes play a very crucial role in the synthesis of fatty acids, which are essential components of cell membrane and serves as energy storage molecules in the form of triglycerides. Now, final one, the fatty acid biosynthesis regulation. Fatty acid biosynthesis is a complex process regulated by several factors to ensure the production of lipids essential for the cellular functioning. The regulation of this process involves intricate mechanism that balances the synthesis and degradation of the fatty acids that are required for the cell's need. Hence, an overview of how fatty acid biosynthesis regulation will be seen here. Enzyme regulation. The key enzyme in the fatty acid biosynthesis is the fatty acid synthase, which catalyzes the synthesis of long-chain fatty acids from acetyl-CoA and malonyl-CoA. Various mechanisms, including allosteric regulation and post-translation modification, regulate fatty acid synthase. For example, citrate is an allosteric activator promoting the fatty acid synthase. Transcriptional regulation, the expression of the genes encoding the enzymes involved in the fatty acid biosynthesis, is very tightly regulated at the transcription level. Transcription factors such as sterol, regulatory element binding proteins, and carbohydrate response element binding proteins play a very key role in regulating the expression of these genes in responsible to the cellular signals. The nutrient availability. The availability of nutrients for fatty acid biosynthesis, such as acetyl-CoA, malonyl-CoA, is a critical factor in regulating the process. These substrates are derived from the glucose metabolism through glycolysis cycle. Insulin and glucose levels can influence the production of these factors that are affecting the fatty acid biosynthesis. Also, hormonal regulation is present. Hormonal regulation such as insulin, glucagon plays a very important role in regulating the fatty acid biosynthesis. Insulin promotes fatty acid synthesis by activating key enzymes and transcriptional factors, while glucagon and leptin have opposing effects, inhibiting fatty acid synthesis to conserve the energy. Feedback regulation. The feedback inhibition also involves the fatty acid biosynthesis is subjected by a long chain fatty acid and other lipid molecules. Excess accumulation of these molecules can inhibit the activity of key enzymes in the pathway, preventing the overproduction of fatty acids. Coming to the summary, in today's session, we have seen the production of straight chain fatty acids, both saturated and unsaturated, as well as the synthesis of odd chain fatty acids. And then we have learned about the subtopics which demonstrate the various complexes of the lipid metabolism, that is understanding the control of fatty acid production with enzymes. And the final one, the crucial and the more study area is that understanding the approaches, how to improve the human health with the help of the regulation of this fatty acid biosynthesis. Thank you.